All right, we're going to take a moment and review something called the cell cycle, which you should have learned about when you were in the ninth grade anyway, or perhaps even earlier. The cell cycle is basically talking about the cell's life from the moment that it is born. It lives a life, it does a job, it's a skin cell, it's being a skin cell, whatever, but then it decides that it must make new skin cells, baby skin cells. And so it is going to divide itself to create new skin cells. So the cell cycle is this cycle of where a cell is living its life, doing its job, growing perhaps in size a little bit, but doing what it needs to do, and then dividing to make new baby cells. Okay? So a big part of this is a topic called mitosis, which is cell division. And we're going to talk about that, but it's going to be a very small part of our talk right now. Our talk is going to focus on the part of the cell cycle that is mostly not mitosis, but on interphase right now. But we do need to understand this idea of cell division, because that is where a cell cycle starts and where it ends. So you started as a single cell created by fusion of sperm and egg, a zygote, very tiny one cell. And you are not that tiny anymore, and you got that way because your cells divided and divided and divided multiple times. They went through the cell cycle many times to create 2 cells from 1, 4 cells from 2, many cells from a few, okay? Cells divide by mitosis for 3 main reasons. If you are a single-celled organism and you're never going to be a multicellular organism, for example, you're an amoeba, then you divide by mitosis for asexual reproduction. You live your life, you say it's time to make baby amoeba, you divide your nucleus, and give your DNA to 2 daughter cells, and now we have 2 amoeba, okay? Asexual reproduction in one-celled organisms, okay? Most larger organisms do not, or at least animals, do not do asexual reproduction as their main way to reproduce. Mitosis is also used for growing, as we've already seen. You went from a zygote to a many-celled organism. We also use it for repairing our tissues, for renewing. If you have skin that's sloughing off, your skin cells make more skin cells to replace those. So tissue repair. So the important thing, though, to remember about mitosis is that we want to create daughter cells that are identical to their parents. We are interested in creating clones. We are not interested in creating all of our little children to be different from each other. We want them, all these little cells that we are creating from a parent cell, to be exactly like a parent cell. I don't want my skin cells to be different from this one. I want them all exactly like this one, thank you very much, because this does its job really well. So we want cells to be identical to parent cells. And this brings us to why I'm inserting this here, because the way we get cells that are identical to the parent cell is that we copy the parent's DNA exactly. We replicate it perfectly. And that's why I put this here, because we're going to be focusing on that process of replication happening in the cell cycle. So, that we've just talked about. With the helicase and the DNA polymerase 3 and all that, we're going to put that into its rightful context in the life of a cell. All right? So, interphase, then, is part of the cell cycle where the cell is spending most of its life, 90%, living its life, doing its job. So if we look at the cell's life as a pie chart, it's going to be spending the vast majority of its time in interphase. And look at this tiny slice of the pie where we're actually going to divide the cell. A very small slice of the pie indeed. Okay? Now, interphase is big, and it's not just all one thing. It's divided into 3 main phases. It's divided into G1, S, and G2. G1 meaning first gap, and that is the first stage of interphase after the cell's been created. G1. We can think of it as growth because that's largely what's happening. S, which is here, and it is called S because of DNA synthesis, creation of DNA happening there. We'll talk about that in a minute. And then the third slice of that pie is G2, second gap. We can also think of that as more growth. The cell's just getting a little bigger. And then there's something called G0, which is not shown here, but I'll show you where that might play in as well. Okay, so G1. The nucleus is quite clear in these cells. The DNA is present, but we can't see individual chromosomes. It's all kind of looks like a plate of spaghetti. The chromosomes, the DNA is wrapped around those histone proteins in a eukaryotic cell, but it's still pretty thin and stringy. And if we looked at all those chromosomes, it would look like a plate of spaghetti noodles. Okay, these chromatin fibers would be hard for us to pick out. But if we did see them, let me go back just a sec. If we did see them, they would be long single strands of DNA double helixes. They would be wrapped around their histone proteins, but if we got rid of the histone proteins, each chromosome would be a one-stranded molecule that was just a double helix of DNA. Okay? Now, during G1, the cell is going to grow, and it is going to prepare to copy those strands of DNA. Okay? It's going to make all the preparations it needs to make sure that it can copy that DNA. Why? Because we may want to give it to daughter cells. Now, at the end of G1, there is something called a G1 checkpoint where the cell kind of checks itself. And if something has gone wrong during G1 and the cell detects it's not going to be able to copy this DNA properly, it'll just shut the cell down. Or if it's a type of cell that doesn't divide a lot, it might shut itself down here and slip out and go into G0, or kind of what is called a resting stage. Okay? And it also would be just doing its job, producing proteins and organelles, whatever the cell needs to do its job and survive. All right, this is that G0 of interphase I told you about. It's when a cell pulls out of the cycle, either because something's wrong with it, and then we got to kind of figure out what to do with that cell, or if it's going to enter into kind of and I put in quotations, permanent non-dividing state. Liver cells, for example, don't normally divide a lot, but if the liver's damaged, they can divide if they have to. We used to thought some cells could never divide again, like nerve cells, but now we know that might not be so true as we thought. But G0 is kind of a non-dividing state, and sometimes the cell can be retrieved from that more easily than others. So here we are. A cell that has just been formed enters into G1, and it grows a little. It makes preparations to enter the DNA replication phase, or S phase. If it's not going to do that, it will slip out of the cell cycle and enter G0, where it just kind of does its thing and does not prepare for division. Now, assuming the cell is ultimately going to divide, we have got to copy our DNA so that we can give DNA to daughter cells. And that is where we would enter the S phase, or the synthesis stage, where the cell is going to replicate its DNA. It's going to copy that DNA. Helicase would be wedged in right here, and DNA polymerase 3 would be going to town on this, and we'd be making copies of our DNA. And so why would we do that? Because this cell has gotten a signal that it needs to make daughter cells, and of course we need to make DNA to give to them. Please understand that the error rate in copying this is extremely low. One mistake per 100 million bases. I couldn't type the Pledge of Allegiance and make just one mistake. I'd make 5. This is amazingly low error rate. But if you're typing 3 billion base pairs, as you would in the human genome, then your cell might make, among those 3 billion bases, 30 errors in a cell cycle. And errors in your copying of your DNA result in mutations, which could be damaging. So your cells do need to go back and kind of correct some of those, even though it's a fantastically low error rate. But there's a source of an amazing amount of variation that could happen. Okay, now please get a picture in your head of what the replicated chromosomes would look like. All right? Please remember that entering into the S phase, we have single-stranded DNA molecules. Okay? Now, this is a single-stranded DNA. Yes, it would be wrapped around the histones, but just pretend we're looking at a DNA molecule. Now, it is going to copy itself, okay? And you've got 46 of these, and they're all going to copy themselves. So this is showing 2 chromosomes here, okay? Well, they are going to copy themselves, and when they do, this single-stranded chromosome we see here is going to get a second arm that is exactly like the first. Here we go. Here it is. There was the first arm. It's going to get another molecule that looks exactly like that. It's going to be genetically identical. That is a double-stranded chromosome. All it means is it's gone through copying. It's copied itself, and the 2 copies are now connected at a point called the centromere. This one copied itself too. It was single-stranded. It is now double-stranded. Once this has happened, we have now copied the DNA, and we can give it to daughter cells. Here is a chromosome that has actually copied itself. Now, this chromosome is a thickened one that we can see. It would not be chromatin, whereas what we are talking about is thin, stringy chromatin that has copied itself. A duplicated chromosome would be the result of the S phase, made up of, as we see here, this whole thing is a chromosome, just as each of these is a chromosome, but when it's like this, we call each arm a sister chromatid, okay? Sister chromatids. So a duplicated chromosome is made up of 2 sister chromatids, and they are attached at a centromere, okay? All right. Now, G2, the cell continues to grow. It makes its proteins, it needs it, but it also gets ready to divide the cell. It gets ready for mitosis. It creates the proteins it's going to need to pull the chromosomes apart and divide the cell. Also, it's going to pass through another checkpoint. We'll call that a G2 checkpoint. And if that DNA has not been copied correctly, we will shut down this cycle and we will figure out what to do with the cell, but we are not going to let it make copies of itself. Again, we don't want to get us a dangerous cell that is mutated in a way that could be harmful to us. So we're going to shut it down. Now, after G2, if we have passed through the G2 checkpoint and everything's okay, we are going to enter into cell division or mitosis, which is actually division of the chromosomes of the DNA. We're going to divide the DNA chromosomes between 2 cells. It takes 4 stages. You might remember prophase, metaphase, anaphase, and telophase, and we'll talk about those in more detail in a bit. Okay? So, that's that. We will catch you in the next podcast. Take care.